Hello learners, welcome once again to our ongoing series on genetics entitled Mendelism and Beyond. In the past two lectures in the series, we have made some very extensive discussion on the mechanism of sex determination in animals. Today, in accordance with the syllabus that we have in genetics, we have already covered with the topics 5 and 6 as you can see on your screens, and it comprises the unit 4. And 7 also we did on the last turn, where we had a very long discussion on how the environment influences sex determination in various clades of animals. Today what we are going to discuss is another very interesting and less understood patterns of sex determination in plants, which comprises the topic number 8. Let us begin by again recapitulating and defining what exactly is sex determination. Actually, sex determination is a biological system that determines the development of sexual characters in an organism. Let me remind you, learners, it was the scientist Camarius who in the year 1694 gave the first description of reproductive organs in plants, and that work was done in Zea mays, that is corn. We can ask ourselves a very pertinent question to begin with. Why does sexual dimorphism exist in the first place at all? Well, of what evolutionary benefit do organisms would gain by developing diploidy and the sexual processes? Well, the answer should be very logical. They have to adjust themselves to the changing environment, and more so, sexual reproduction will allow much more variation to take place in the system, and it's very obvious, more variation would mean a better food for the process of evolution. But then, what is better about the combining of gametes to produce a new generation of offspring rather than just remaining with the vegetative propagation? The answer to this query again needs some logic, because if we are going to have more of the reproduction by gametes, it would combine beneficial mutations and it would also help in removing the deleterious ones. So that means the incorporation of the good ones and the elimination of the bad ones or undesirable ones is also a routine makeup of evolutionary process. Let us summarize the modern theories which have been given in terms of sex determination. As you remember, learners, we have discussed many of them in the reference of animals. The first is the chromosomal theory of sex determination. Obviously, it would revolve around the chromosomes and the genes which are present on them. The male and the female gametes would fuse together, syngamy would take place, and we would have the formation of a zygote. And as you know, these gametes are going to be specialized cells, and they would have a haploid number of chromosomes, which after fusion would give rise to a diploid individual. And then there are two types of chromosomes, namely the autosomes and the sex chromosomes. The autosomes are responsible for determining the phenotypic body characters, whereas the chromosomes which are present in a diploid organisms, we would find that there will not be any difference in the location of genes in the male and the female, and the number and morphology, in other words, would remain conserved in males as well as the females. On the other hand, there are sex chromosomes which are also called as allosomes and the heterosomes. They are the ones which are going to determine sex, and they are of different types. Then another modern theory is the genic balance theory of sex determination, which you remember we discussed was given by Calvin B. Bridges in the year 1925, and we had a detailed discussion on sex ratios of Drosophila melanogaster. So here in this case, the sex of an individual is going to be determined by a balance between the genes for maleness and those for femaleness present in its genetic makeup. So it's a kind of it's a balance. So therefore, the individuals which are going to carry the genes for both the sexes, actually it would be the ratio between the X chromosomes and the autosomes which is going to govern the development of male as well as the female sex. This ratio is called as the sex index ratio. As you can see on your screens, learners, the sex index ratio, which is abbreviated as X by A, here A would mean not just the autosomes but the number of autosome sets, and the numerator would be X chromosome number. So the values and their expressions are different. For example, if X by A ratio is 0.5 or less, it would differentiate into a male. If the ratio becomes say 1 or greater than 1, then it becomes a female. And you must have noticed in your books that we have, say, if it is greater than 1, it could even be a super or a meta female. And finally, if the X by A ratio ranges between 0.1 and that is greater than 0.5 and less than 1, then it would mean that the individual would be an intersex. There is another type of sex determining mechanism, learners, which is the famous haplo-diplo mechanism. We've also discussed this mechanism with reference to bees, wasps and various other insects. Here actually the females, they are developed from a fertilized egg. So if they are developed from a fertilized egg, that means they are diploid. The males develop by a different method of parthenogenesis, that means without fertilization, and this process leads them to become haploids. So that means we have two ploidy levels in the females and the males respectively. And we also discussed the aspect of arenotochus development. So this is very common, as I said, in insects like bees, ants and wasps. And then there's another theory, which is the cytoplasmic theory of sex determination, which is also very primitive because it is seen in bacteria like Escherichia coli, where we find that there is an extra chromosomal DNA which is called as the sex factor or the F plasmid, and it is this which is responsible for sexuality. So what really happens is that when the strains of E. coli are conjugating, because conjugation is one of the methods for sexual transfer of the genetic material, now the donor cell transfers its F factor into the recipient cell, and finally the F minus cell becomes F plus cell. Or a situation could arise that the F factor integrates with the DNA of the donor cell, and when it does so, then it transforms into what is called as a high frequency recombinant or HFR cell, and this can then transfer its genetic material onto the recipient cell. There's another set of theory which is the nutritional theory, and this was proposed by Sharp, and this says that it's not only the genetics which dictates the future sex of an individual, it also depends upon the nourishment of the gametes. Obviously, size would also be a consideration in that. So many plants would also show this type of a mechanism. For example, the size of the egg is going to also determine the nature of the sex of an individual. The small egg would develop into a male, and whereas the large egg would develop into a female. This is very natural because the female has to have enough nutrition to nourish the next or the coming generation. So every zygote, in other words, is going to contain all the responsible genes required for the development of sex, and it is the nutrition or the other environmental conditions which are going to decide as to in which direction the sex of an individual would go. And this is better exemplified in another set of theories, which is the environmental theory of sex determination, which suggests that environmental factors, they would play a direct or an indirect role in determining the sex of an organism. We classify such sex determination mechanisms as ESD, that is environment induced sex determination, and this is the most direct means of a variation in sex ratios of the progeny. You must have seen that there is in a population, the number of males and the females may not be mathematically exactly equal. One of these sexes would be more. So that means the expression of a particular sex, that is a male or a female, is going to be determined by internal as well as the external environmental conditions. Because under certain situations we would find that a gene for a particular sex gets inhibited. Now, once it is inhibited, that means the other sex would take precedence, and it would express itself, and the process may even get reversed, because if the conditions or the environmental conditions change, then the whole story is going to be reversed. There's another set of theories which are studied under the hormonal control. If you remember, learners, we had a very detailed discussion in certain animals. The hormones, they are playing a very important role in the differentiation of the sexual characters. We had a long discussion on Bonilla, how it fell, and when it was alone, it was a female, and when it fell on the proboscis of the female, there were certain hormonal secretions which inhibited the femaleness, and the male progeny came through. And likewise, the free martin intersex cattle, we had a discussion on that, where the twins, in case of cows, one of them would be male, which would be normal, but the female would have external genitalia of a female, but then it would be sterile, and it would have defunct ovaries or non-functional ovaries. So in that case, this female would be called as an intersex. Now the question is, we know a lot about all these systems which I have just mentioned, but then why are we still studying them? The point is that understanding the different types of sex determination systems is very critical to the survival and evolution of sexually reproducing organisms, because it is not a static entity. These sex determination mechanisms are being modified over a evolutionary period, and we need to have a knowledge of the lineages also. So that means, although we know a lot about the existing systems, but sex determination systems can rapidly evolve from one system to the other. So we need to be abreast with all these changes that are taking place. And moreover, we are always directing the situations of sex determination to humans, to ourselves, because it is very apparent. But then there are organisms which are other than humans, and here the genetic sex of offspring might not be revealed at all, but then it is equally important. Let us take the example of crop plants. The sex of an individual plant is very important for food production, because in many crops like melon, squash, strawberries and grapes, we would find that it's only the female flowers which are making fruit. So what kind of flowers are there? Are they joined with the male flowers? Are the flowers bisexual, hermaphrodite, perfect, whether they are monoecious or dioecious? So we have all these types of variables. One thing is very clear. Learners, you should remember that the life cycles are going to depend on sexual differentiation. And when we are talking about a multicellular organism, one must distinguish between primary sexual differentiation, because this will involve the gonads, where the gametes are produced, and it has nothing to do with the other external appearance. That would come under the secondary sexual differentiation. That would involve the overall appearance of an organism. So that means we need to appreciate and understand the connection or the relationship between the primary sexual differentiation and the secondary sexual differentiation. On your screens, learners, you can find that there are different types of the sex determination mechanisms which we have to study in plants. They would be environmental, chromosomal, and when we are talking about chromosomal, then there's a huge array of variation, because the chromosomes could be homomorphic. They could be, when they are homomorphic, that means the males. They could be heterogametic, or as we saw in case of birds, they would follow a XY female and XX male system, which we called as the ZWZ system. And then we have what we call as a heteromorphic chromosomes, where we have an active Y. And then imagine the plants also show certain systems where there is a sex determination based on the balance between the X chromosome and the autosomes. So that means a balance theory. And then apart from it, there are various mechanisms which we would study. They are the genic mechanisms, and there are some which involve just a single locus and various multiple loci. So let us begin with the environmental sex determination in plants, which is also a very interesting aspect. As we know, learners, the environmental sex determination would involve where the gender is decided after conception. That means, depending upon the environment, the sex of an individual is not genetically fixed. And till now we found that our literature says that the environmental sex determination has been studied extensively in angiosperms, in flowering plants, because we find that the plants there are sexually labile. That means they are flexible. But then when we say that there is environmental sex determination, that means what we are indicating is that here the establishment of sex is by a non-genetic cue. Say for example, nutrient availability, which is experienced within a period of time after fertilization. So it is very different from the genotypic sex determination, because the genotypic sex determination is going to establish sex at fertilization by genetic factors. Say for example, like the sex chromosomes, or if there are no sex chromosomes, then certain genes, which we will also study subsequently. But then environmental factors are going to influence the determination of sex during the development of sexual maturation. Say for example, light intensity, photo period, temperature, availability of nutrients, and even certain pheromones which are emitted by the surrounding plants or animals. They are also going to influence the sex of an individual. So once the sex is determined under a particular environment, it is fixed, and then it will not be switched again. Please remember, learners, that if a light period has changed, or it has differentiated a particular plant into a male, then by changing the environment you cannot change it back into a female. So that means we should really be very clear that environmental sex determination is different from some forms of say sequential hermaphroditism, where we find that the sex is determined flexibly after fertilization throughout the organism's life. That means you could change it into a male and the female and back to male in one life cycle. If we were to just look at the screens, we look at a tuft of a moss, and in this case we find that the green cushion is comprised by the gametophytes, which are upright, and then the sporophytes are also visible as the upright brown structures. Now, there are so many sporophytes, that means so many fertilizations have taken place, that means there must be so many male and female plants here, whether they are dioecious or monoecious, that we have to understand. If they are monoecious, then obviously they would be self-fertilizing, or if they are dioecious, they would have then the fertile males and the females separately. Now, having dioecious plants is a bit rare, because we find in terms of the population genetics also, the females are usually out competing the males in mosses. So that means we find that the flexibility is less as far as the genetic system is concerned, and after fertilization we find that once we have fixed a particular sex, it would remain so. When we consider a sex determination mechanism in mosses, there are some very interesting facts which have come to light. Learners, you will be surprised to know that the sex chromosomes were reported in Spherocarpos as early as in 1917 by Ellen. But then most of the gametophytes in mosses, we say, are going to be asexually reproducing, and they could be even males or females or even hermaphrodites like ferns. But then moss gametophyte could either be monoecious, that means in monoecious what we call as the homothalic, or it could be dioecious, same like we find in case of vascular plants. But then most important dioecious species of mosses, they show a unique genetic sex determination mechanism which is called as UV, sex chromosome determining system. Here the UV does not mean ultraviolet. Here UV means the U chromosome and the V, sex chromosome. So that means if there are two of these sex chromosomes, then what do we expect? We expect that the chromosomal sex determination should result in production of 1:1 production of male and female spores. As you find, say for example, in humans we have the X and Y sex determination mechanism. But then we can always detect in mosses, that is the females, as I said before, they are going to outnumber the males in most of the species that have been studied so far. Well, it could be possible that nutrient is one of the factors, and the nutrient concentration is known to influence sex determination. For example, in the dioecious moss, Bryum argentium, we find that if say for example auxins and gibberellins are given to this moss, it would have more of male plants, and if cytokinins are given to it, then the plants are showing more of femaleness or more of female plants. Somewhat similar results have also been reported in the liverbird Rixia crystallina. Here we find that the treatment given by gibberellins and ethereal, which is a source of ethylene, they enhance antheridial formation, that is, they enhance maleness. They are androgenic, while the auxins and glycosel, they would promote the development of archegonia. That means they are feminizing. And there are, on the other hand, certain monoecious moss species like sphagnum. They are also exhibiting environmental sex determination during early development with low light, low pH, low nutrient availability. All these factors are going to promote male development. And if there is high pH, high nutrient availability and good light intensity, it obviously is going to result in the differentiation of females. So, it is believed that what could be the reason behind the interaction of the hormones? If auxins and gibberellins are present, there must be some compounds which are, say, similar to antheridogens that we are going to study in ferns, and both male and female individuals are going to vary in terms of the presence or the number of their antheridia and archegonia, depending upon what is the balance between these hormones. Likewise, what we find is that there is a concept called as cluster effect, that is too much of crowding. It is seen in a monoecious species of tetraphis, where we find that the sex expression is sensitive to density, and if male shoots are dominating, that means they are very dense in a tuft, and perhaps more antheridogens are going to be released due to the cluster effect. And naturally you would agree that the female mosses have a competitive advantage, as they can be maintained by minimal sexual and high vegetative reproduction. So that means there is a differential survival rate and spatial segregation of sexes in the female dominated populations. On the other hand, the environmental sex determination is labile, it is flexible, and it naturally shows a dynamic development of females or males in different environmental conditions. We find that the lability or the flexibility of this sex expression is in many plant taxa, but in case of homosporous pteridophytes, it is actually a rule rather than an exception. And most of the fern species, as you know, are homosporous. That is, they have only one type of spores, with the exception of course, salviniales, where we have Salvinia, Azolla, and many others. But then they lack sex chromosomes. So that means every fern spore would have the capacity to make a male or a female or a hermaphrodite gametophyte after its germination, but then it would depend on the environment. On your screens you would find that there is a fern gametophyte which is immature and it is at an asexual stage, and it may now, what are the cues which are given by the surrounding environment, the direction of the sex would follow the other ones. A similar situation occurs in the horsetails, that is equisetum, which is also called as the puzzle grass, and the wild populations tend to be again female biased because there are more of females. So what really happens is it depends on the photon irradiance. On your screens you would find that if the photosynthetically active radiation is between 24 and 28 micromoles per square meter per second, then most of the in vitro grown gametophytes will develop as females. But then If it was less, then they would be in the form of males. We can also talk about the addition of sugars, which are going to give rise to change in its nutrition, and it would also promote male development. On your screens you would find the cluster effect, and therefore we summarize that these particular lower plants, they are used to crowding pH and sucrose concentration, and they would modulate their sex expression. In our next lecture, we would continue this discussion by taking examples of other plant clades. Thank you.